fourth participant is Jordan Miner from our Graduate School of Biomedical Science and Engineering.
Imagine you've just been diagnosed with early-stage breast cancer. What are the immediate emotions running through your mind? Scared, stressed, anxious? Now what if I tell you at an 88% chance of living another 10 years if you do absolutely nothing? Would you seek treatment, or would you take the risk? This is a problem faced by 20 to 25% of all breast cancer patients, and it's not an easy one to solve. Currently, we have limited methods to detect if an early-stage breast cancer will progress to a more deadly form or will settle idle, causing no harm for the patient. Most early-stage breast cancers will never progress to become invasive. However, the thought of living with cancer and all the uncertainties that surround this problem are a mental burden for patients. Therefore, the standard of care is typically mastectomy or breast-conserving surgery due to its unpredictability. Initiatives are starting to emerge to combat this problem we know as overtreatment. However, data-driven biomarkers are still desperately needed to separate slow from aggressive cancer. Without these biomarkers, patients may receive treatments that do not optimally balance risk versus outcome. My solution? Collagen. Collagen is the most abundant fibrous protein in our body and has recently been identified by other scientists as a biomarker for cancer progression by observing changes in its structural pattern. I want to take this one step further and understand how the cancer cells themselves influence the collagen. Cells interact with collagen through receptors on their cell surface. I hypothesize that by understanding which of these receptors is more active in cancer progression could lead to development of new biomarkers. To study this, I am using 3D models of human breast cancer cell lines that range from early stage, as seen on the left, to late stage, as seen on the right. All of these cell lines are derived from one patient, allowing the disease progression to be studied under a common genetic background, increasing reliability. I am selectively removing each of these cell surface receptors and then observing any changes to cellular invasion, as seen here, as well as collagen organization. We've already found that one of these receptors could be a unique biomarker for breast cancer patients. Ultimately, I aim to make the decision of whether to treat or not to treat early-stage breast cancer a little easier on patients, improving their life, health, and well-being. Thank you.